Hi everyone, I'm Michaela Crawford, and today we'll be covering jumping genes, a discovery made by Barbara McClintock. For this presentation, we'll specifically be covering Barbara McClintock herself, what jumping genes are, how they were discovered, and different types of transposons. Before we begin, let's get to know a little bit about Barbara herself. She was an American scientist and cytogeneticist born on June 16, 1902 in Connecticut. She majored in biology at Cornell University in 1919 and received a BS in 1923, a master's degree in 1925, and finally a PhD in botany in 1927, having specialized in cytology, genetics, and zoology. She then accepted a teaching position at the Uni of Missouri in 1936 and finally received several awards, including the 1983 Nobel Prize in Physiology and Medicine, before dying in 1995 on September 2nd in New York. She is, of course, famously known for her work in the genetic structure of maize, where she discovered more than 50 years ago that some genes could be mobile, and found a chromosome breaking locus, which is the location of a particular gene within a chromosome, that could change its position within a chromosome. Thanks to her discovery, we now know that jumping genes constitute more than 65% of our genome and 85% of maize genomes, which confirms that transposons are more widely spread among eukaryotes. This confirmation from maize geneticists led to a wider appreciation of Barbara's original discovery, and so after around 30 years of withholding her research due to the cold shoulder of the public, McClintock finally received awards for her work and remains the only woman to have an unshared Nobel Prize in Physiology. Despite her poor relationship with her mother, McClintock's field of choice, biology, actually makes sense due to her close relationship with her father, who was a medical doctor and physician who allowed Barbara to spend her time however she liked, which was usually inside reading her books. It was actually her father who had to talk Barbara's mom into letting her attend college, which her mother, Sarah, was against for fear that no man would want to marry Barbara. This was actually a common family concern for daughters during this time. She invented the field of cytogenetics more than 20 years before Watson and Crick identified the structure of DNA. She was the first to actually prove that genes were physically located on chromosomes by also finding that chromosomes swap some genetic info by crossing over when sex cells are formed. This is something we learned about around a year or two back and is also the explanation for why siblings with the same parents look different. As you know, she made this discovery based on her curiosity of the color patterns of corn, like purple corn or corn with spots and speckles, which for a while were thought to be mutated white corn. Her research came to explain that this color difference only happened when parts of the genome moved from one place to another until sometimes landing a gene for color, hence the gene jumping. Barbara was driven to study transposable elements on chromosomes in an attempt to test Roland Adam Anderson's 1910 research on the occurrence of purple or brown spots on supposedly colorless white kernels in maize, and judge his hypothesis that the spots were due to unstable mutations. So, she bred maize plants to vary in kernel color, then took samples from the corn and dyed them to look under a microscope using staining techniques to enhance the contrast in microscopic images, and observed the chromosomes. She bred maize plants carrying chromosomes that resulted in a recessive brown phenotype called BZ, with maize plants that held chromosomes coded for a dominant white phenotype called colorless. The offspring turned out white with some brown spots, instead of all white, following the normal theories of inheritance that Barbara attributed unexpected variation to a chromosomal breakage where a chromosome lost one allele and called the phenomenon dissociation. Her hypothesis ended up being that the kernels carrying three alleles must have lost both their dominant colorless allele and their dominant purple allele due to the breakage, resulting in the white with brown spots. She also argued that the amount of brown spots on the creamy background depended on the timing of the breakage during development. Now, what exactly are jumping genes? Jumping genes, also known as transposable elements, TE, or transposons, are DNA sequences that can change their position within a genome. This sometimes causes or prevents mutations or changes the cell's genetic identity. Barbara managed to isolate two genes, what she called the controlling elements, which control the genes responsible for pigmentation. And since Barbara McClintock's discovery, three basic types of transposons have actually been identified, and they are class 2 transposons, miniature inverted repeat transposable elements, also known as MITEs or class 3 transposons, and retrotransposons or class 1 transposons. Class 2 elements are simply segments of DNA that move from one place to another using a cut and paste mechanism, like scissors. Basically, all of these elements encode an enzyme called transposase, which acts to cleave the ends of the transposon and frees it from its initial location in the genome. Transposase also cleaves the target sites for element insertion, and once the transposon is bound into its new position, gaps that are left into the DNA sequence are filled in through nucleotide synthesis. This class of transposon can range in length from 1,000 to as many as 40,000 base pairs. In contrast to class 2, MITEs have short lengths generally ranging from about 400 to 600 base pairs by a stretch of 15 base pairs that occur at each element invertedly as mirror sequences. Unlike some types of transposons, MITEs don't seem to encode proteins, and most insertions of these elements happen in euchromatin, which is a form of chromosomal material that contains most active genes. A regulatory function of MITEs were proposed thanks to this, and it's that some microRNAs are derived from MITEs. Despite this knowledge, the actual mechanism for how these elements move in the genome still isn't understood. Retrotransposons are a highly unique group of transposable elements and form large portions of the genomes in many eukaryotes. Retrotransposons also function by a copy and paste mechanism, and so they leave behind the original copy and generate a second copy that's inserted somewhere else in the genome. This process results in the insertion of repetitive sequences of DNA throughout the genome, and is the mechanism responsible for the widespread of transposable elements in many higher organisms. The first step in retrotransposition occurs when the transposable DNA is copied into RNA. Then the RNA segment jumps to another location in the genome. However, the RNA must be copied back into DNA by an enzyme called reverse transcriptase in order to be inserted into the genome at the new site. There are several different types of retrotransposons, including long interparsed nuclear elements and short interparsed nuclear elements, also known as LINEs and SINEs, or lines and signs, and about 20% of the human genome is actually made up of LINEs. The simplest kind of transposons only contain a copy of transposase with no additional genes and behave like parasitic elements and usually don't have a known function associated with them that are advantageous to the host. They typically do have additional genes with them, and they can be antibiotic resistant, which usually happens when an infecting bacterium gets a plasmid that carries a gene encoding resistance to one or more antibiotics. These resistant genes are typically carried on transposable elements that have moved into plasmids and are easily transferred between organisms. So once a bacterium picks up a gene like this, it can grow in the presence of the antibiotic. This is usually promoted by indiscriminate use of antibiotics. Interestingly enough, the actual functions of transposons are still unclear. For a long time, they were called junk DNA because they seem to serve no purpose, or they were called selfish DNA because they only copy and amplify themselves within the genomes. Apart from genetic diversity, in rare cases, transposons are associated with genetic mutations or chromosomal rearrangements that cause disease in humans. This is because disease typically arises from the insertion of transposons into particular regions of genes that are involved in regulating gene activity. For example, insertions near promoter regions can actually lead to overactivity of genes like cancer. In other cases, the site where a class II element is cut out of a gene and not repaired properly, which can result in mutations that interfere with gene regulation and cause cellular dysfunction. Diseases like hemophilia and Duchenne muscular dystrophy and several more are associated with repetitive DNA arising from retrotransposons. I'd like to thank you guys so much for listening, and I actually did prepare this little activity for anyone who is willing to participate, where you either state the most memorable facts you heard, or, since I did mention two, state the university that denied Barbara from teaching. Thanks again. My references are on the last slide.